Hello and welcome to this MedChemNet interview with Dr. Kevin Reid. Kevin joins us as part of our focus on drug discovery for diseases in the developing world. Kevin is the head of drug metabolism and pharmacokinetics at Dundee's drug discovery unit based in the UK. And he is accountable for the strategy and coordination of DMPK support to all DDU projects, using in silico ADME models together with a range of in vitro and in vivo tools to ensure rapid identification of quality hit series and timely progress through to pre-clinical candidates. Kevin has extensive experience of early phase drug discovery, lead optimization, project leadership, and preclinical development gained from over 25 years in the pharmaceutical industry and his time at the DDU. Kevin, thank you for joining us.
Thank you, Peter.
If I go straight into the questions then. So to start off with, could you please provide an introduction to Chagas and your research conducted at the DDU?
Sure, yes. So Chagas disease is a tropical parasitic disease. It's caused by the protozoan parasite Trypanosoma cruzi, which is spread mostly by the blood-sucking bugs of the Triatominae family, and often referred to as a kissing bug. Now, they're called kissing bugs because when they emerge from the crevices and walls in the nights, they go down to the inhabitants that are sleeping in their bed and tend to feed off their faces. So quite an unpleasant creature. But anyway, the disease can also be spread through blood transfusion, eating contaminated food, and from the pregnant mother to the fetus. Now, this disease affects around 8 to 10 million people, mostly in Mexico, Central and Southern America, but it's increasingly spreading to other areas such as North America, Europe, Japan and Australia, mainly due to migration and the fact that the majority of people infected with this disease are asymptomatic. So when a person's infected with Chagas disease, there's an acute stage which occurs shortly after the initial infection, can last up to a couple of months. And as I said earlier, it's usually asymptomatic or unrecognized simply because the mild symptoms that the person has are not unique to Chagas disease. So things such as fever, body aches, muscle pain, loss of appetite. Very rarely some people will die from the acute disease, a very, very small percentage because of severe inflammation. And the acute phase can also be severe in people that are having weakened immune systems, such as those with HIV. So after those 8 to 12 weeks, the chronic phase of the disease develops when the immune system is activated, and this results in a dramatic reduction in the parasite load in the body. And then people that have chronic Chagas disease remain asymptomatic can remain asymptomatic for decades. But 10 to 40% will eventually develop some life-threatening heart or digestive disorder such as cardiomyopathy or megacolon, and this is where the real problem starts, and it's causing approximately 8,000 deaths a year as it stands at the moment.
Okay, and with that at the moment, what are the current treatment options and why is it that they fall short?
Well, the current treatment options are very, very limited, unfortunately, in that there's only two very old nitroheterocyclic drugs available, benznidazole and nifurtimox. They're both effective in the acute stage, but very limited in their effectiveness in the chronically infected people. So the other problem is that the side effect profile of both is very poor. About 30% of people taking benznidazole will get allergic dermatitis, as example. Because of the very poor tolerance, this leads to compliance issues. So some people will stop treatment mid-course, which could explain some of the limited effectiveness actually of these drugs in chronically infected patients. We've also got resistant strains of the Trypanosoma cruzi out there to these drugs, and actually access to these drugs has been an issue in the past, although that actually is starting to improve. So really, it's very clear that there is a very clear need for new, safe, well-tolerated treatments for this disease, and this is very much why the drug discovery unit at the University of Dundee, is now funded by the Wellcome Trust to carry out, small molecule drug discovery for Chagas disease in a fully collaborative partnership with GlaxoSmithKline in Tres Cantos in Spain. our focus in this disease is very much on identifying novel active chemical series and then progressing those through lead optimization to deliver preclinical candidates that GSK will then take through the formal GLP preclinical development phase, and then in all likelihood will then partner with the Drugs for Neglected Diseases initiative as we move into clinical trials with these new medicines, or new potential medicines, I should say.
Okay, excellent. And then following on from that, do you have any ideas on what form a novel therapy for Chagas might take, or are there any treatments you feel particularly optimistic about?
This is a very challenging question, actually. I mean, at the moment, unfortunately, the drug pipeline for Chagas disease is currently empty. The only new potential treatments that are progressing through preclinical development at the moment are additional nitroheterocyclic drugs that continue to carry a toxicity risk because of the reactive nature of these chemotypes. But there's also a very promising oxaborole that's in preclinical development with the Drugs for Neglected Diseases initiative. So we're keeping a close eye on that. But at the moment, with our own programs, our current efforts are really on trying to understand what a non-reactive small molecule chemotype really needs to look like to be able to deliver sterile cure in the patient. Because the challenge with this particular parasitic disease is the parasites are very extensively distributed in the body, including sort of immunoprivileged conserved sites such as the brain, the ovaries, the testes, as well as actually in the lymph. So actually us designing a molecule that has all the compound properties that are needed to distribute well to all of these sites really is going to be a major challenge, and further research is needed with a range of tool compounds to explore what indeed is actually needed in a novel, small, non-reactive molecule. And indeed, we may find that actually combination therapy in the future with pairs of molecules may be the only way forward with non-nitro type drugs.
Okay. And so my next question, would you be able to tell us a bit about the two new assay methods developed and utilized at the DDU for Chagas?
Yeah, so yeah, I think with the ones you're alluding to. So when we started our drug discovery efforts for Chagas disease, the drug discovery pathway for this disease really wasn't clear at all, and we've had to evolve and develop key assays and modify the screening cascade for progressing molecules as our own knowledge has built up. And one of these key assays, I think, is that you're alluding to, is a static cidal assay, as it's clearly very important that we prioritize our medicinal chemistry efforts on only those compound series that are delivering rapidly cidal compounds against the parasites. Now, the second assay, or the key assay that we've established, has really evolved in response to evolving clinical data. So there's been two azole drugs in recent times, posaconazole and ravuconazole, although ravuconazole was administered clinically as the prodrug. But these unfortunately failed in clinical trials for Chagas disease. And now both of these, the mechanism of action in the parasite was through CYP51 inhibition. So consequently, not surprisingly, it's put into question this mode of action as a viable drug target for Chagas disease. Now, the majority of our early drug discovery in Chagas disease is through phenotypic screening, so screening compounds against the whole parasite. So we don't know what the target is. So it's important for us, it's certainly important for us to assess whether our active hit series are actually acting predominantly through that pathway which is under question, or is it something else, so that we could prioritize our medicinal chemistry appropriately. So what we did was utilize our DMPK expertise and in CYP450 inhibition assay development to actually create a high throughput assay for Trypanosoma cruzi CYP51 inhibition, and then use this high throughput assay to screen all our active hits that are arising from our phenotypic screening campaigns to see whether that activity has been driven by that particular mechanism, and then prioritizing our chemistry away from that mode of action because of the question mark that currently exists over it from the clinical data that's come through. And this has proved very, very valuable within our programs. There was a remarkable enrichment of our hit series to that particular mode of action so we had to remove all of those series from our current efforts. And indeed, we're also using this assay to support the activity of other groups in the Chagas drug discovery community to ensure that they're working hopefully on novel modes of action as well.
Oh, fantastic. And so as you alluded to there, employing those assays, you identified 2 orally bioavailable lead series, one of which entered lead optimization at GSK. Are there any sort of preliminary findings or anything extra you'd be able to share with us about that?
Yeah, I mean, there's a number of active compound series that we have that are not working by CYP51 and have good oral pharmacokinetics that we're currently exploring all of these at the moment to see if we can deliver sterile cure in relevant disease models. And this is happening currently and over the coming months, and hopefully that's going to help us answer some of our hypotheses as to the compound distributional properties that are really going to be needed to deliver a sterile cure for this disease. Now, the original lead series, unfortunately, we've closed, and again, I think that was related, as we were learning more, to the actual mode of action. It was a much slower killer, and it was more CYP51-like in terms of its biological profile, but actually was not a CYP51 inhibitor in its own right. So again, I think the mode of action is going to be incredibly important within this disease as well. So again, that's added to our learnings, but we're in good shape with a number of hit series with good pharmacokinetics at the moment that we're now exploring to see whether we can achieve a full sterile cure at the moment in disease models.
Okay, excellent. I'll be keeping my fingers crossed.
Yes.
And then for my final question, in an ideal world, if you had unlimited funding, what sort of research would you want to carry out and why?
a lot more research into this disease at the moment. We're very much still in the early stages and understanding, what we need in a molecule that's going to deliver cure in this disease. So really, we need a lot more research in understanding the pharmacokinetic and the pharmacodynamic relationship for delivering cure in relevant models of disease for a small molecule that doesn't work by the reactive mechanism that the current successful drugs are. And then, we work with one particular strain of T. cruzi. Is this distributional pattern we see consistent across the strains? So some of the biological understanding needs to be addressed more. Are there dormant forms even, something that's not easy to answer at the moment? And if there are, that would clearly make the mode of action for some of our drugs more critical to deliver sterile cure, because it's much harder to hit some of the mechanisms on a much slower growing or more dormant form of the parasite than a faster growing parasite. And then, of course, ultimately, because the pipeline at the moment is so poor, we really do want to fill the pipeline with a large number of drug candidates with new modes of actions, get a lot more drugs into clinical trials, proof of concept, and then use that clinical data to back translate the current drug discovery pathway that's been set up and the various animal models of disease that are out there as to whether they are truly translational or not, and refine the drug discovery pathway. At the moment, there's very limited clinical data in which to build the pathway, so we're really reliant on getting some more things through to then back translate that we're doing the right thing. And then thirdly, in terms of what we would do, is obviously most importantly, I think, is around trying to identify surrogate markers for parasitological cure. The current clinical readout, which is PCR, it gives you a good early indication of treatment failure, but not treatment efficacy. And as I mentioned in the introduction for the disease, this can take decades to manifest itself in chronic disease, if at all, in some patients. So to get an accurate measure of treatment efficacy currently could take many years. So a quicker feedback mechanism for back translation as to whether you truly have achieved sterile cure clinically would be highly, highly valuable.
Oh, fantastic. That's a great answer. As I said, that was my final question. So I'd like to thank Kevin for his time, and also to remind our viewers that MedChemNet is on Twitter, @MedChemNet, and we'd be absolutely delighted to hear your thoughts on today's interview. Kevin, thank you once again for joining us.
Thank you.